Mono something? Mono. Mono.
So, welcome students. I hope you all have joined now for the another second session of analytical techniques. Good. So, in order to go further, so we have just discussed today in the morning about various techniques in electrophoresis, and before that we have discussed about DNA isolation and PCR, and we have done various virtual workshops for them. And we also had a virtual workshop about agrosgel electrophoresis, and we just discussed last time in the first session about your pulse field gel electrophoresis PFG. Now we continue further. Uh, this is your further type in the zone electrophoresis, that is your thin layer electrophoresis. In this, studies can be carried out in thin layers, so of silica, alumina, or Kaishgua. So its advantages, we could get really good resolution consuming, and it's widely used in combined electrophoretic chromatography studies in 2D of a protein and nucleic acid synthesis. Further, there is a cellulose acetate electrophoresis, but these are not so important, as in we are not using them mostly, so I'm not taking too much details over them. So just a little bit touch towards them, each of them. So cellulose acetate electrophoresis, they contain 2 to 3 acetyl groups per glucose unit, and absorption capacity is less than that of copper paper, and it's give sharper bands, provide a good background for staining glycoproteins mainly. So some of the advantages, disadvantages, application is an alternative to paper electrophoresis, and its analysis could be good for clinical and biological protein samples like albumin, globins, globulins. And comes your moving boundary electrophoresis. In the moving boundary electrophoresis, it's allow the charged species to migrate in free moving solutions without any supporting media. So you have one side anode and cathode. This is the one very nice beads here. So you have ascending boundary, initial boundaries there, and this is the descending boundary. Your buffer is inside. So according to the charge, your protein molecules will disperse. So that's why it's called moving boundary electrophoresis. So inlet our sample here and they start to move according to their things. So it consists of a U-shaped glass cell of rectangular cross sections with electrodes placed on each of the limbs of the cell. And sample solution is introduced at the bottom or through the right side arm. And the apparatus is placed in the constant temperature bath at 40 degrees Celsius. And detection is done by measuring refractive index throughout the solution. So biologically active fractions can be recovered without the use of denaturing agents. A reference method for measuring electrophoretic mobilities and minute concentrations of the samples can be detected. And disadvantages are costlier, elaborate optical systems are required, and applications that is used for to study the homogeneity of macromolecular system and analysis of complex biological mixtures. This one is quite important, capillary electrophoresis. So that I was using with my friend. So that person was working during his PhD on Prions diseases. Prions, the neurodegenerative disorders. So in this capillary electrophoresis, main principle behind the electrophoresis is that the charged molecules will migrate towards the opposite poles and separate from each other based on their physical characteristics. And they have been grown to become collections of range of separation techniques. And in this, basically, we perform electrophoresis in buffer filled narrow bore capillaries, normally 25 to 100 millimeter in internal diameter, and we give a high voltage of 10 to 30 kilovolt, and the inner diameter of these capillaries are 550 micrometer, and the length is around 0.5 to 1 meter in the length. That's some concept. You have a cathode anode. They are like the boundary electrophoresis binded together, but we added some computer to it in order to generate data that could be help with the help of a light source could be injected. So capillary electrophoresis. Maybe we can have one virtual workshop for that. If something is there. If not, then we can. Some instrumentation is there. We will see. I think this one is there.
In this video, we will introduce you to the new capillary electrophoresis system, CAPL-205, developed and manufactured by Lumex Instruments. CE can be a cost-effective addition to a lab, taking many routine analyses off chromatography and therefore reducing operational expenses. With CAPL-205, you get a qualitative and quantitative analysis in a great variety of application areas, such as water quality control and environmental monitoring, food testing and agriculture, pharmaceuticals, and biochemical research. CAPL-205 can detect all classes of analytes, organic and inorganic, ionic and non-ionic, polymers and monomers, optical, cis, trans, and constitutional isomers, and others. All of this is done with one instrument. Its efficient electrophoresis separation technique provides fast determination of multiple components in one run. The analysis time is typically just 5 minutes. Another advantage of CAPL 205 is low analysis cost, thanks to very low sample and reagent consumption, in addition to the affordable consumables. Unlike expensive HPLC columns, capillary cassettes with automatic capillary rinse are affordable and easy to use. They allow you to run many different analyses without changing. You can easily switch between protocols just by exchanging the cassettes and loading the necessary files. The instrument has a high-capacity autosampler of 59 positions for standard centrifuge-type tubes, which is a cost-effective solution for both high-throughput labs and laboratories with just several samples per day. CAPL 205 is run by the powerful and user-friendly ELFO-Run software that controls the instrument and acquires and processes the data. Another advantage is the wide library of validated standard operating procedures that can be run on one instrument. Moreover, Lumex Instruments participates in the development of national and regional standards on a regular basis. The instrument comes with the relevant analytical protocols and all necessary reagents according to the customer's request. Let me show you the determination of anions in water as an example. The sample pretreatment usually consists of just filtration, dilution if necessary, and centrifugation. Then we place the centrifuge tube containing the pretreated sample directly into the autosampler and start the measurement. The analysis is done. CAPL 205 determines the concentration of 6 inorganic anions in water within just 3 minutes. You can save the obtained report on your PC and print. CAPL 205 is a universal instrument capable of performing any types and modes of CE analyses. It provides you low-cost, simple, fast, and accurate measurements in a great variety of application areas. Please visit our website to find out more about the instrument and method and make the right choice for your lab.
So that was about capillary electrophoresis. Then comes the electroosmotic flow. This is what happened in that. So you have your, as we have discussed, negative electrodes and negative ions, and you will have also positive ions. So the positive ions will attach to the negative ions, and in between there will be positive and negative. So they will be flowing, giving a osmotic flow, electroosmotic flow. Will be distributing from a node to a cathode. So that's the diagram, the small volume of sample that is moved into one end of the capillary, and the capillary possesses through a detector, usually UV absorbance detector, and the opposite end of the things. So you can collect the data from here. And there is isotechphoresis. This is done for the development of potential gradient. This is based on the principle of moving boundary electrophoresis. In this, you have leading electrolyte with higher mobility than the analytes and trailing electrolytes. So we can see in this example shown here with 3 particle classes with different charges being separated and pre-concentrated by electrophoresis, and after the separation they are concluded at the constant speeds. So like 1, 2, 3. So ABC. So in this, we will have our one electric field, slow trailing electrolytes. So we have this capital T and mixed with our sample S1 and S2. And also we will also have a fast leading electrolytes, that is your L. So when we apply electric field into this ions electron migrates through the microchannel according to the electrophoretic mobilities. So sample ions will start moving through the trailing electrolyte but cannot overspeed the fast leading electrolytes. Consequently, they focus at the interference. So by the end, what you will see, your small one S1 and S2, they are somewhere in between. They got separated. Your trailings will be on the left and your leading will be on the right. So based on that, your isotoc process works out. And there's immunoelectrophoresis. In the immunoelectrophoresis, we are having the antibodies that are produced by immune system in response to the foreign micromolecules. In this, basically, your antibodies are playing role. Antibody and antigen complex are formed. So when the electrical potential is applied to study of antigen-antibody reactions, it is called immunoelectrophoresis. And the antibody are electrophoretically separated. Antigens diffuse towards each other, resulting in the precipitin arcs. So here these are the arcs that are produced, unpaired arcs. But once the antigen is added, so they got separated and they attach on the antibody like this. So in this, we usually have 2% agar gel medium. The antigen mixture is applied into the small circular wells cut out of the agar. And after the initial separation, the antibody mixture is then introduced into a narrow slot in the gel. And during this period, the antigen components diffuse readily towards outwards and towards the diffusing antibody, and precipitation takes place in elliptical arcs as related antigens and antibodies diffuse into one another. So some of the advantages, that is the spreading of bands is minimized due to the application of the application field. And disadvantages are the carrier ampholytes generally are used in relatively high concentrations, and high voltage power up to 2000 volt is necessary. Power in the vicinity of 2 to 50 volts. Maybe we can have, this is quite important aspect of immunoelectrophoresis because of its it is being sometimes used in the immunology labs where you have vaccine technology. So there they are using this technique. So this is rocket immunoelectrophoresis.
Rocket immunoelectrophoresis. This technique was discovered by Laurel in 1966. Principle of this technique is immunodiffusion and electrophoresis.
This is not good. So we want to see exact that how it is happening. That should be our goal is.
Hello friends, today we will learn the technique of.
I think it's not possible to see this technique practically. It's not available, unfortunately. Maybe this one. Mostly people are giving lectures over the YouTube.
I am going to discuss today is
No. So unfortunately not. Okay, then isoelectric focusing. So I will skip this part because we will discuss about isoelectric focusing in our chromatography. where we will discuss how 2D gel electrophoresis is done in details. So we can skip this thing. And this is a 2D. We will also discuss about 2D gel, like how we make 2D gel. So all these parts will be discussed in details in the next lectures of your gel electrophoresis. So we are done with this electrophoresis part of today. Okay. Now, in continuation with the electrophoresis, let's check with the further techniques in electrophoresis. That is your northern blotting, southern blotting. So various blotting techniques. So in this blotting techniques, basically we will discuss about how can you separate your DNA, RNA, and protein using your southern, northern, western blotting. So southern is for DNA, northern is for for RNA, and western is for protein. So we will discuss all these techniques one by one. Southern western is the combination of to find the interaction between your DNA and protein, northern western RNA and protein. Then there is a dot blotting and zoom blotting. So let's start with the first technique, that is southern blotting. So this southern blotting is basically done in order to check target DNA fragments that have been size fractioned by gel electrophoresis. And this technique was invented in 1975 by E.M. Southern. So this principle in this technique was to exploit the property of radiolabel probes with a single stranded DNA. And if we want to detect the presence of specific sequence in our mixed DNA samples, then we will accordingly design the probes which will have complementary sequence to our target sequence. So in procedure, called as southern blot, in this what we do is we get the DNA sample and we cleave them with the help of restriction enzymes like restriction endonuclease, and then we run upon the gel electrophoresis. And the double-stranded helix of each DNA fragment is then denatured into the single strands by making pH of gel basics, and the gel is blotted with a sheet of nitrocellulose, and then transferring some of the DNA strands to the sheet. The next, a probe consisting of purified single-stranded DNA corresponding to specific gene is poured over the sheet. So any fragments that has nucleotide sequence complementary to the probe will hybridize with the probe. If the probe has been labeled with 32P, it will be radioactive, and the sheet will show a band of radioactive where the probe is hybridized with the complementary fragments. So here you have your gel with all the nucleic acids on top, and we mark them with radioactive markers with specific sizes, and then we make this transfer of our gel particles over the nitrocellulose membrane with the help of stacks of power paper towels, and in center is your nitrocellulose membrane and is a gel. And afterwards you took the gel, you put the nitrocellulose paper and contains nucleic acid printout, and afterwards what you do is you have your paper, right, your nitrocellulose membrane, and you add correspondingly radioactive labeled nucleic acids, and then we can watch them with the help of radiograph, R2 radiograph, and look for our hybridized nucleic acids. So in this southern blotting, basically we can have the information about the whether the particular gene is present or not, or is there any rearrangement happened during the cloning. We get to know about the chromosomal genes and probe sequence, so it has many advantages. And the uses could be to detect cancers and genetic diseases, to determine the number of copies of a particular DNA sequence, used in discovery and gene mapping genetic patterns in the organism DNA. So let's watch how we do this southern blotting technique in practical way. So there will be subtitles coming below. There will be no sound. So just follow them. So it will be more about practical aspect, how we do it in the lab.
There will be.
Yes. So that was about southern blotting. Now let's discuss about northern blotting, which is quite similar though. It's not so different, but the initial steps are quite different from that. So in the northern blotting, we are not working with the DNA. We are particularly interested in the RNA in the northern, and they're separated according to the size. And we also hybridize with the probe, like the way we did in the southern blotting, and we look for the sequence target of this. So while working with RNA, we should take lots of precautions to avoid the degradation of RNA by ribonuclease, that is the RNase, which is found in our skin or glassware. So this has to be taken extra care. And we also add DEPC also here, which inhibits your ribonuclease activity. And also we bake at high temperature to destroy all the ribonuclease activity in this. So same, we isolate RNA, then we put run over the gel, we transfer to the nylon membrane, then hybridize with the buffer. Then RNA blots are usually put with a complementary DNA fragments. Then the unbounded probes are moved by washing. Then we check over the R2 radiograph with X-ray film. So the important information that we get from RNA blotting, that to get to know about the particular gene, in which tissue is it expressing at various stages of development, if the expression changes under different conditions or not. The quantity of mRNA is also being checked, whether a genomic DNA probe has regions that are transcribed. Then uses of northern blotting, it's allowed researchers to determine gene expression patterns. And this indicates a myriad of practical applications, allowing researchers to compare patterns of gene expressions in cells of tissues. And the northern blotting analysis can be used to detect cancerous pancreatic cells and tissues. And also it's enabled the scientists to know the functions of unknown proteins, and it's enabled the scientists to detect the size of RNA also. So it's alternate splice products using the probes with partial homology. So let's watch how it is done practically. So let's do the next part of Western blotting. Western blotting is mainly done for the proteins which are analytical techniques used to detect specific proteins in a given sample of tissue homogenates or extract. Sometimes their effort was immunoblots, which we use gel electrophoresis to separate your native or denatured proteins with the help of your 3D structures of proteins. So this name was Western blot given by the technique by Neil Burnett, which play Southern blot was also with his name. So he developed technique for DNA detection earlier actually than the advent Southern. So in this technique, protein sample is electrophoresed on SDS page and then electro transferred onto the nitrocellulose membrane. The transferred protein then is detected using some specific primary antibody and secondary enzyme labeled antibody substrates are provided. So in this procedure, in this technique, all the samples of proteins are separated on the basis of their molecular mass using SDS or 2D electrophoresis. And electrophoresis move the proteins from the gels onto the nitrocellulose membrane where proteins adhere. And to detect the specific proteins, an antibody to that protein must be available. And the nitrocellulose membrane itself has many non-specific sites that can bind proteins. So the primary antibody is added into the solution and binds to the proteins of the interest. An antibody complex is detected using secondary antibody that has a label attached to it. So often the reporter enzyme that we use, such as alkaline phosphatase, is linked to the secondary antibody, and addition of lumophos or X-phos to the blot allows the detection of the protein band. So this is the technique that we use in Western blotting. So we have nitrocellulose membrane with attached proteins to it, and then we add a primary antibody which is get attached to this specific protein, and then we add secondary antibody which will attach to the primary antibody, and then with the help of chemiluminescence we visualize them. So information that we get from Western blotting is size of proteins, expression amount of protein, and the uses of Western blottings are useful, very much helpful in confirming HIV infections, HIV test, Western blots to detect anti-HIV antibody, and further, and so on. So now let's watch a very nice detailed um, video about Western blotting explaining the technique in detailed manner.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You will be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay one fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper. Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer, and then discard the wash buffer. Add another 50 milliliters of wash buffer to the membrane, and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
Very useful technique that is being widely used in the lab. Are used around 70 to 60% this technique. Secondary body bache, it's mainly, so it's a horse secondary antibody, donkey antibody, or it's just basically attaching to your primary antibody. That is, it is being the animal from that it has been carried out. So that's why it's attaching to specific it, and then after attaching the secondary antibody, it is we can able to watch our horsadics, the chemiluminescence agents will be able to attach on the secondary antibody. They are not able to attach on the primary antibody. If we don't add secondary antibody, we cannot see them. It's not visible for us later on. So that was southern blotting. Then there's a southern western blotting. As I discussed, it will be dealing with the DNA and protein interaction, and the northern western for the protein and RNA interaction. And the dot blotting is a technique which was identified, which is a modified version of western blot technique, but it differs in a way. Though we are look for the protein analysis in this, But in this, we do not separate the protein samples using electrophoresis, but instead we spot these onto the membrane and hybridize with the antibody probe. We just spot these things and then it's go for further, but it's not highly used. Mainly your Western blotting is being used properly. Then comes your last blotting technique, that is your zoo blotting technique, that is in which it is a technique based on the principle of southern blotting. And in this genome of any organisms, so there are two regions. There's coding and non-coding regions. The coding region is the most interested part for the researchers, which gives genetic information for specific protein. But the problem is that most of the DNA that are non-coding, the question is how to identify coding regions in large amount of non-coding DNA. So zoo blotting helps in this in order to distinguish between your DNA from non-coding regions. So during evolution, the base sequence of non-coding DNA mutates change rapidly, whereas the coding sequence change much more slowly and can still be recognized after millions of years of divergence between two species. So therefore, DNA is extracted from a series of related animals such as a human, monkey, mouse, hamster, cow. The sample in this DNAs were cut off with suitable restriction enzymes, and the fragments are run on a gel electrophoresis, and then we hybridize with particular DNA and then check for things. So here we have in this probe on the left hand side non-coding binding to itself, and the right hand side other one probe with coding DNA. So we took 4 different species in this technique. Probe is segment of human DNA that may not be formed from the coding region. So blotting is carried out as a usual. On the left, the only hybrid seen was between probe and the human. Therefore, related sequence were not found in other species, and probe is probably non-coding DNA. So in this example on the right, the probe binds to related sequence in other animals. Therefore, the piece of DNA is probably from the coding region. So that's it of um, coding your blotting sequence. So we are done with electrophoresis, various types of electrophoresis, various types of blotting techniques. We have discussed the DNA isolations till now. We have discussed the PCR. So on that note, let me have just 10 minutes break. We meet at 12:10 or 12:20. And then we start with our next session of yeah, we do a little bit about DNA immunoprecipitation, that is your chip sequencing, chromatin immunoprecipitation, and then later on we start with our chromatography part. Okay, let's wait for some time. This is being read out. So chromatin immunoprecipitation, that is your chip principle. That's quite important technique also. Basically, in chromatin immunoprecipitation, what we do it is that we check the cross-linking of your cells. That you have a DNA, and we remove this DNA from the cells, and then we want to check the cross-linking with which transcription factor or which protein is it being playing role. So in order to do so, we add antibody to it and then immunoprecipitate with that antibody. Then we wash the antibody away. Then we can find the cross-link reversal and then we purify this DNA and then quantify with the quantitative PCR, and then we check for DNA and protein analysis that which DNA and protein are coming together. That's a quite apt summary for this chromatin immunoprecipitation. So in this case, not only proteins interact with one another, they can also interact with DNA. So in chromatin immunoprecipitation, that is a chip technique that determines whether a protein of interest interacts with specific DNA sequence. So this technique is often used to study the repertoire of sites on DNA that are bound by particular transcription factors or by histone proteins to look at the precise genomic equations of various histones modifications such as acetylation, phosphorylations, or methylation. And how does this chip works? So chip can be used to examine the presence of protein DNA interactions at steady state or quantify changes in interaction at specific phases of the cell cycle or follow treatment of interest. So protein and associated chromatins are temporarily cross-linked in live cells or tissues and sheared using enzymatic digestions or sonications to yield 300 to 1000 base pair fragments of DNA. And the protein of interest, along with any associated DNA fragments, is immunoprecipitated from the cell's debris using a specific antibody. The cross-link is then reversed and DNA fragments are purified, and the amount of eluted DNA can be assessed through quantitative real-time PCR using primers flanking the genomic locus of the interest. And the DNA amplification is an indication of enrichment in binding of the proteins of interest. So in the modified versions of chip techniques, we will discuss one by one. So one is chip on chip, which is done for genome-wide analysis of protein binding sites using microarray analysis of purified DNA. Chip sequence, this is also genome-wide analysis of protein binding sites. Then this we use not microarray, but we use deep sequencing of purified DNA fragments. Native chip, which omits the cross-linking step and mainly uses micrococal nuclease digestion in order to cut DNA from the histone linkers and to identify the DNA target of histone modifying proteins. Then chip exo, this was done with the exonuclease digestions to identify to make the better resolution of protein binding sites, and it's up to single base pair. Then CHIA-PET, there is chromatin interaction analysis by paired and tag sequencing. So this technique is the combinations of principles of chip with chromosome conformations, which capture 3 carbon to detect long chain chromatin interaction mediated via protein of interest. An iChip, this is the indexing first chromatin immunoprecipitations, a high sensitivity technique that reduces the number of cells required for chip experiments by initially barcoding total cellular chromatin. And chip, this is engineered DNA binding molecule mediated chromatin immunoprecipitation. This technique actually using CRISPR-Cas9 system, for which in 2020 in physiology a ma'am got the Nobel Prize winner who have identified it. So this is a guided RNA complementary to the desired genomic region, is expressed in the combination with tagged enzymatically inactive Cas9 protein. Chip is then performed using an antibody against modified Cas9, and this technique can help evaluate cis transinteracting chromosomal looping events. And RIP chip sequence, RIP chip, as a similar technique used to analyze protein RNA interaction. So some limitations are also there of chip, that these are molecular biology techniques. Chip is not without its own limitations, so it always has some limitations also, such as the presence of protein at a given genome locus. It cannot be determined functional significance of a protein binding at that region. Cross-linking may additionally include proteins that transiently interact with the DNA or DNA binding proteins, have no functional significance. Similarly, DNA interactions of proteins with short residence times may not be fully captured. Additionally, interacting proteins may mask the epitope of the protein of interest. Finally, the chip technique is extremely dependent on the quality and specificity of the antibody and may not discriminate between different DNA binding isoforms. So some important factors while doing so, some troubleshootings, that is while doing so garbage in and out, that is the quality of chromatin generated in chip can make the difference between uninterpretable results. So like sonication could break the chromatin and down into shorter fragments, and sonicated for too long could also denature the proteins. So this could lead to various bad results. Also, keeping chromatin on ice for all the time could make your reactions bad. Also, overheating, denaturing could lead to bad things to your protein. Antibody need to be validated, that what kind of antibody is using, is the epitope is fine, that is going on. Further, cell number consideration, the cell number should be in optimized level. They should not be higher or lower. If it is not in the adequate amount, then it will be a problem for us. And cross-linking issues. Cross-linking is a time-sensitive step and should only be performed for short durations. And prolonged cross-linking can reduce antigen availability and efficiency on the chromatin sharing steps. Then appropriate controls are required. IgG, input controls, positive controls, negative control locus. These has to be maintained. So now let's watch one video about this chip.
Come in closer, let me whisper something in your ear about ChipSeek. Yeah, StatQuest. Yeah. Hello, I'm Josh Starmer and welcome to StatQuest. Today we have a gentle introduction to ChipSeek. Note, this StatQuest is based on the gentle introduction to RNA-seq, so watch that first unless you're already totally down with RNA-seq. Okay, we've got a bunch of cells. Inside each cell, there's a nucleus. And inside each nucleus, there are a bunch of chromosomes. Let's focus on the chromosomes. Specifically, let's focus on these chromosomes. Chromosomes are made out of chromatin. Chromatin is made out of DNA, plus histones, a type of protein, plus other proteins that we'll talk about some other day. They're not that important for understanding ChipSeq. DNA wraps around the histones to package DNA, and the packaging can regulate gene transcription. Depending on how they are modified, histones can activate or repress genes. Lots more can be said about chromatin and how it's packaged, but that's for another day. Today we just need to know that chromatin is essentially DNA wrapped around histones. To simplify things, let's just use this big blue line to represent chromatin from here on out, but remembering that it's made of DNA and histone proteins. And let these big brown arrows represent genes. And let this green circle represent a histone that allows transcription. And let this red stop sign represent a histone that represses transcription. In a cell, all kinds of proteins can bind to DNA. This mustard-colored thing might promote gene transcription of this gene. And this pinkish-colored thing might repress this gene. And who knows what this green thing is doing? We can use ChipSeq to find out. ChipSeq stands for chromatin immunoprecipitation combined with high-throughput sequencing. It identifies the locations in the genome bound by proteins. That's the most important thing in this StatQuest, so I'll say it again, in bold. ChipSeq identifies the locations in the genome bound by proteins. For example, say like we wanted to identify all the regions in the genome bound by the green thing. The first thing we do is use formaldehyde, or something like it, to glue all the proteins bound to the DNA together with the DNA. This means that all of the DNA-bound proteins, including the ones we're not interested in, are glued to the DNA. The next thing we do is cut the DNA up into small, approximately 300 base pair, fragments. Then we isolate the protein we're interested in. In this case, we're interested in the green thing, using an antibody. The black star represents an antibody that is attached to a bead. Then we isolate the proteins bound by the antibody and wash everything else away. Then we reverse the formaldehyde glue by warming up everything. Then we isolate the DNA by washing away the proteins, including the histones. Now that we see how to isolate DNA that is bound by a particular protein, let's take a step back. So far, the example has focused on just these chromosomes. But the process, glue proteins to DNA, cut up DNA, bind proteins of interest with antibodies, isolate antibodies, unglue and wash away proteins, applies to all the chromosomes in the cell. And it is usually applied to a pool of 6 million cells, give or take a few. So we end up with a lot of DNA fragments from a lot of cells. Then, just like with RNA-seq, we prepare a sequencing library by adding sequencing adapters to both ends of the DNA fragments. Then, just like RNA-seq, we PCR amplify the library, check the library concentration, sequence, filter out garbage reads, and then align the high-quality reads to a genome. That is to say, if this is the genome, the first read might come from here, a location on chromosome 2. The second read might come from here, a location on chromosome 1. The third read might come from here, another position on chromosome 1, etc., etc., etc. Ultimately, we get a long list of genomic coordinates for all the reads, usually between 50 and 100 million reads. And we can use those reads to create a genome browser track. These are genes and chromosome positions in the mouse genome, MM10 to be exact. This is the track that we created for our ChIP-seq reads. A lot of reads map to this region, and relatively few reads map to these other regions. This track was made from a control experiment. The control track was made by taking some of the input chromatin from the original ChIP-seq experiment, and, without using an antibody to enrich for any particular protein, ungluing all the proteins and washing them off. Then sequencing, aligning, and converting into a track. In summary, the control track uses some of the same input chromatin for the ChIP-seq experiment, but doesn't try to enrich for any particular protein binding. We use the control track to verify that a high concentration of reads in the ChIP-seq track is due to a protein binding there, and not because a lot of reads map to a repetitive region. Statistically significant peaks are usually represented on genome tracks by rectangular bars. We could then compare peaks for the same protein in different cell types, like lung versus kidney. Or, if we didn't know the specific DNA sequence that this green thing bound to, we could guess that it is a motif found in all of the peaks. Here's a motif found within the peaks that indicate where the green thing bound. The large letters are more frequently associated with the green thing than the little letters. We can also try to determine the functional role of the green thing by looking at where it binds relative to the genes. Here, we see that the green thing binds near the start of a gene, so it might regulate that gene in some way. Anyway, those are some of the things you can do with ChIP-seq. In summary, ChIP-seq identifies the locations in the genome bound by proteins. Bam! Hooray! We made it to the end of another exciting stat quest. If you like this stat quest, and want to see more, please subscribe. And if you want to support StatQuest, please click the like button and consider buying one of my original songs. Okay, until next time, quest on.
So that was about chip sequence. Now I take you to the another part of, because I want to start with electrophoresis, but we still have 20 minutes left. So if I start that, that will be in between left. So maybe I can do something else in these 20 minutes. Okay, we start. So now we start with the protein part, that is your chromatography. So how do you isolate protein? How do you quantify it? Then how do you purify it? Okay, everything has been there now with you. So these are the various famous Nobel Prize winners on electrophoresis. That is your John Porter Martin in 1952, your Syringe in 1952 for partition chromatography, and Arne for his research on the electrophoresis and absorption analysis. So all these people very famous. Then John B.F.N. 2002, they work on the mass spectrometry and sedimentation. Tanaka, Swedberg, and people working on the organelle fractionations, Albert Claude, Christian de Duve, George Pallade, in their respective years. And Frederick Sanger, he found his two times Nobel Prize winner, one in 1958, one in 1980. Once first at the work of the insulin at the University of Cambridge, then MRC Laboratory of Molecular Biology in United Kingdom in 1919, so 1980. So like this, so on the basis of finding the base sequence of your nucleic acids. So let's see how we isolate first of all a protein. You can isolate your protein with the help of first step as a disrupting or enzyme cells, fractionations, crude separations, homogenates into the fractionations. So in the homogenization, first you can do it with the Downs homogenizer, sonication, osmotic shock, or French press. In the fractionation, you can do it with the help of differential centrifugation, ammonium sulfate precipitation, or functional precipitation. So here in this diagram, we can see in order to isolate your specific part of your organelle, you can do the use of centrifugation. So let's say you have everything homogenized form. Homogenized form here, and you centrifuge for 500 G at 500 G for 10 minutes. So you have the supernatant and the pellet, and the pellet will include your all nuclear fractionations. Then you again centrifugate the double like 10,000 G for 20 minutes, and in the pellet you will have all the mitochondrial fractions. Then again you make 100,000 G for 1 hour, then you will have all microsomal fractionations in this pellet. And the cytosol, all the soluble proteins will be there in the cytosol part. So there are two types of centrifugations to separate the molecules based on their mass, density, and shape. That is differential centrifugation and rate zonal centrifugation. And that differential centrifugation here, samples poured in the tube, you will have more dense particles and less dense particles. And in the rate zonal centrifugation, you will have layered. We put the mixture of your layers of your large particles and small particles over the sucrose gradient. So in the left, you do the normal centrifugation, and you will have the palette. You can separate the palette and remove the decant liquid into the container. Whereas on the other side, in the rate zonal centrifugation, once the centrifugation is done, you will see your mixture is separated according to the different density. So you can separate them using the pipette easily. Then protein isolation, that is centrifugation. Um, it is at 3000, we do it at 3000 G for 5 minutes with large organelles, and we have 10,000 G for 10 minutes, in which we have mitochondria and inclusions bodies. And we also do with the 40,000 G for 30 minutes, so like membrane, plasma membrane, tonoplast, microsomes, secretory vesicles, peroxisomes, and 200,000 G for 16 hours, like large protein molecules, complexes. And at 400,000 G for 24 hours, your protein could be for 50 Dalton. So in sedimentation velocity technique, basically what you do is you have low density solution and high density solution, yeah, and you put your samples into this rest tube, and then you put your layering sample on the top, and they got separated according to their different sedimentation coefficient, which could be calculated with this formula. So every Swedberg velocity technique has S values different for different protein. For trypsin, it's 2.5. So for carbonic anhydrase, 3.23. Conclave A as 3.8. So like so. And sedimentation velocity technique according to the density and according to sedimentation coefficient. So we have this graph separating RNA, DNA, ribosomes, chloroplasts, nuclei, mitochondria, soluble proteins, viruses, microsomes, all being separated into this coefficient. So they will be all separated with this particular coefficient and this particular density. You can separate your things that you're looking for. After this, getting your protein, after the isolation of protein, now it's high time that we should have the quantification of your protein. So one method is Lowry method, in which we can determine from 10 microgram to 1000 microgram per ml. But in the Bradford protein determination method, this technique is quite simpler, faster than the Lowry method, and it's subject to less interference. And the Commercy blue G250 dye appears to bind most readily to organyl lysyl residues of proteins, and this specificity can lead to variations in the response of the assay to different proteins, which is the main disadvantage of the method. So how we do this Bradford assay? Let's have a video. This will be the last thing that we will do now.
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So we stop it here, our lectures. Yeah. And then if you have any question, you can ask me now. And then we meet at 3 o'clock today. Students, I have shared you one link here. Please give your honest feedback on this workshop, or if you want to go for further workshop, contact Vatsal sir. So we have many workshops ranging from MBBT, proteomics, medicinal biochemistry, principle of genetics, plant biotech, bioinformatics, drug discovery, genomics, proteomics, enzyme technology, analytical techniques, molecular biology. So we are running many, not only for 2 days, but we are also running for 1 month in detailed manner for 1 month technique things. So please open to yourself and meet the people and let be in contact with them. If you have no further questions, so I will switch off from here, and then we see us at 3 p.m. Yeah, for the further input with this.